The first speaker will be Dr. Onikepe Folarin.
Yeah, thank you very much. And let me say good evening from Nigeria. I am Onikepe Folarin. I'm an associate professor at the Department of Biological Sciences, Redeemers University. And I'm also the Deputy Director of African Center of Excellence for Genomics of Infectious Diseases, also based at Redeemers University. I'm making a presentation on behalf of African Centers of Excellence for Genomics of Infectious Diseases. And my topic is to share what our platform is. So it's a platform for genomics research and capacity building in Africa. Next slide, please. So African Center of Excellence for Genomics of Infectious Diseases is a World Bank funded center, and we're based in Redeemers University in Ede, Osun State. How did we start? I'm going to start with the quotes made by the director of the center just about the time the center was to be set up. And he said, I quote, "Our vision is to set up an environment where African scientists can express their God-given talents. But it is not just about the science. Rather, we envisage the creation of a stimulating environment that will foster interaction between scientists in a way that will provide solutions to the obliterations of infectious diseases." This was the dream and the vision of the director of the center, that is in the person of Christian Happy. Next slide, please. So what brought about the establishment of ACEGID is the fact that we know that the Human Genome Project, following its completion, has revolutionized the way we approach healthcare and research. This we see in its potential to transform clinical care, disease surveillance, understanding of infectious diseases, drug discovery, and vaccine discovery, which is our focus for this presentation today. Next slide, please. So I'm going to talk about ACEGID. How did it come about? It's a coalition of a long-standing research and training partnerships based in Africa, that is including Redeemers University, University of Ibadan, and also some clinical sites or institutions, that is IRA Specialist Teaching Hospital. And then across the region, we also have the Kenema Government Hospital, and then we have the Sheikh Anta Diop University in Senegal. Our international partners include Harvard University's Broad Institute, Tulane University, Cogenics, and then other collaborators who are more of funders are Wellcome Trust, US Department of Defense, and others. Next slide, please. And that is taking us to our mission. Next slide, please. Our mission is in twofold. We have education and research mission. For our education, it is about capacity building. We hope to be able to create a vibrant academic and research environment that is free of outside influences and that transcends national boundaries and ensures the conduct of relevant, responsive, ethical, and high-quality translational genomics-based research on health in Africa. And our research goal is to characterize fevers of unknown origin through microbial metagenomics. This we have been doing in the past 8 years, and the intercept that it creates, next slide, please. The intercept that it creates is such that between education and research, we end up having capacity being built in the area of genomics so that we can have genomics teaching and sequencing lab, diagnostics core facility, and sustainable science careers for our young scientists. Next slide, please. And to a layman's language, what we hope to do at ACEGID is to create the next generation of African pathogen hunters. What we want is in such a way that these pathogens don't hunt us down, but rather we hunt them down. And going forward, following the establishment of ACEGID, the first thing we did was to see how we can narrow down the turnaround time of genomic sequencing. And this we did successfully by converting a 30 days process to about 10 days process. And that's what is giving us the power to be able to carry out real-time genomic surveillance, as we have seen in the case of COVID-19. Next slide, please. I'm going to give us a brief history of how we started, what we have been doing before now. And the first one that actually brought us to limelight was the Ebola virus disease outbreak in 2014. And SGID was at the forefront of it, where we took up the responsibility of detecting and diagnosing the virus in disease cases. And apart from that, apart from detecting the virus, we also carry out the genome sequencing of this virus within the country. This singular real-time detection.
Rubbish car. Let me ask you.
Can you mute
Hello,
your mic?
You guys rubbish collectors, yeah? Yeah. What's your number? You got a card?
Can you mute your mic, please? So a singular act of being able to detect this virus in real time was one of the main success stories of Nigeria in early containment of the disease in 2014. And we did not only stop at detection and carrying out the genome sequencing, we used the genome sequencing information to develop a rapid diagnostic test kit that can detect the presence of the virus in 10 minutes in any suspected cases. This has been approved by WHO and US FDA for emergency use. Next slide, please. In our bid to continue to hunt pathogens, we were able to discover 2 novel rhabdoviruses in blood of healthy individuals from West Africa, specifically in Edo State. And we named those viruses Epomavirus 1 and Epomavirus 2 to show where they were first identified. And what we discovered is that these rhabdoviruses have been in existence because after carrying out seroprevalence analysis, we discovered that most of the population in that environment have been exposed to this disease. And so maybe that is why we do not have an outbreak, because there has been some level of exposure. Next slide, please. We also do a bit of Lassa fever virus research. And in 2018, when the outbreak seems to be extraordinary because of the large cases that was reported, we carried out real-time genome sequencing of the outbreak of samples, where we sequenced about 90% of the Lassa fever virus then. And what was our outcome? It was that we found out that the Lassa virus exhibits extensive genetic diversity, and it is still a case of rodent to human transmission. And next slide, yes. So apart from even detecting the Lassa virus, we also went ahead to carry out metagenomic analysis of the blood samples obtained from this patient. And we found out that there were many other pathogens in these individuals, ranging from HIV, GB virus, yellow fever virus, trypanosomia brucei, and of course Plasmodium, which we know that is highly endemic in this environment. Next slide, please. So we realized that there is power of local genomic capacity based on the demand for us to be involved in some other analysis. One significant one was the presence of white powder at the US Embassy in 2017 and 2019. So we were called on to do the analysis, even though the powder did not contain any pathogenic substance following our metagenomic analysis, but it is costing us a lot right now in this country, because right now the US Embassy is not open for drop box again. We also took part in the monkeypox detection, and then we went further to try and identify the Ebola virus and Lassa virus in their natural reservoir, that is the fruit bat and the rodent reservoir. You will agree with me that there's a growing demand for the local genomic capacity. In 2018, there was a yellow fever outbreak, even though initially it was not thought to be, it was just showing as an hemorrhagic fever. And the samples were tested for Lassa fever, but it was negative for Lassa fever. And we carried out real-time metagenomic analysis, and we were able to identify that what was causing the hemorrhagic fever was yellow fever. The details of that was conveyed to the Nigeria CDC, and they immediately announced an outbreak of yellow fever. But aside of us being able to detect the yellow fever, we were also able to identify that these yellow fever strains were closely related to yellow fever strain identified in Senegal as against those ones that were earlier identified in Nigeria in the 60s. Next slide. Thank you. So going on to the business of today, the SARS-CoV-2. This broke out in China in December 2019. The first case was reported in Nigeria on February 27th, 2020. And immediately it was identified to be COVID-19. We received the sample on the 1st of March 2020, and we carried out our test and the metagenomic sequencing, and this sequencing was released on the 6th of March 2020, even though the result came out on the 4th of March 2020. And the genomic information that we found there confirmed the case that the virus is actually SARS-CoV-2 virus and is also related to the virus identified in Europe, as is expected. I'm sure we all know the origin of the index case. Next slide. And so following that first report of COVID-19 genome, which came from her lab at ASGID, we have, between then and March 2021, we have been able to identify 55 different variants which spreads across the country, as you can see, ranging from FCT Abuja, Oyo State, Lagos, and all the other states in the country. Next slide, please. One of the variants that was reported of concern in UK, B117, was also identified by our lab, which was identified on the 14th of March 2021, even though these samples were collected sometimes in December 2020. And this variant was found to be spreading across the country even faster than the early variant of the virus that was first identified. Next slide, please. As you can see, this B117 lineage, you can see it's spreading, and we carried out this analysis in Osun, Oyo, Edo, Lagos, Abuja, Kwara, and Ebonyi. And you can see that it has almost taken over the initial variant of the virus. Next slide, please. In addition to seeing this particular variant, we also looked at mutations that were identified in the variant, in the UK variants, to see how similar these variants that were detected in Nigeria was like. And we found out that it was also about the same deletion, HV69 to 70 deletion as in the UK, as well as the Wuhan sample, and also has the N501Y mutation. And so this confirms that we also have the B117 virus circulating in Nigeria. And in addition to the B117 virus, we also identified a new lineage, B1525 variant, which was identified in Nigeria on the 11th of February. And this particular variant had, in addition to all the previous mutation identified on B117, also has additional mutation and additional deletion. And so far, its origin has been located in Nigeria. And more research is ongoing to understand the transmission dynamics of this particular virus. Next slide, please. Next slide, please. So we do not only carry out metagenomics analysis and stop there. Please, can you go back? Thank you. We do not only do metagenomics analysis and stop there. One of the things that we want to do is, we know that infectious disease has three major concerns. That is vaccine development, drug development, and diagnostics. If we're able to find solution to all of these three, then we know that we'll be able to control and manage these pathogenic diseases whenever they come out into the human population. And so one of the things we have tried to do at ASGID is to be able to develop some rapid diagnostic test kits that will enable us to be prepared for any outbreak that may come. Next slide. And this is in in a panel that we call SHINE, which is Streamlined Highlighting of Infections to Navigate Epidemics. Our main focus at SGID is to be pandemic prepared or outbreak prepared. And so we are trying to develop a panel that contains some of these pathogenic organisms and that we will be able to identify them in human sample. So this panel or the SHINE panel to help us to identify this pathogen in 5 minutes if it's individual samples, and if you are pulling samples for high throughput, we will be able to identify these pathogens in roughly 1.5 hours. Next slide, please. We are actually validating this SHINE assay on the SARS-CoV-2 samples using the saliva and the nasal swabs, and these have been tested in 500 Nigerian RNA sample. And so far, the sensitivity has been high, especially in samples with high viral load. And we're working on making an improvement for samples with low RNA input. And this we have seen based on, sorry, can you please go back? We are also looking at the probability of using this SHINE in samples that are non-invasive. So we're talking about saliva and urine, and this will actually make diagnosis very easy. One of the main advantages of this SHINE is that the development is very rapid, lasting for about maximum of 6 days, and it would be rolled out for validation and immediate use. Next slide, please. So the long-term goal of SGID is to be able to develop deep-rooted scientists in the field of genomics that would be actively involved in genomic surveillance of pathogens, so that, like I said, these pathogens will not hunt us down. So we want to be able to develop a group of scientists that can co-create and build capacity in the field of genomics with sustainability and openness of mind to be able to find a solution to the challenges of infectious diseases through research and partnership. Next slide, please. So I'm going to end this presentation with also a quote from Christian Happy, shortly after Nigeria was declared Ebola-free. And he said, I quote, "Our hope is that next time this happens, we will be able to perform deep sequencing right on African soil." And I think at this point, we have actually been able to achieve that, because the very first SARS-CoV-2 African genome was actually done at SGID in Nigeria and in Africa. Next slide, please. So I would like to acknowledge all our team members and our funders, and most especially the Nigerian Magazine, for giving me this platform to make this presentation on behalf of SGID. Next slide, please. For more information, you can visit our website. And last slide, next slide. Thank you for your attention.
Well, thank you very much, Dr. Folarin, for the presentation. Very, very encouraging. Encouraging in the sense that you are doing real work. Your work you started before, and the work that you had done, you built on, even with the SARS-CoV-19. And you've talked about, like what you do in terms of education, building capacity, research. You've talked about that you hope to have a next generation of African pathogen hunters. And also that now you are turning around time of genomic sequencing from 30 to 10 days process. So they are very, very encouraging. And I know there are a lot of people that would want to ask you questions later on. So I think we'll keep it till the Q&A bit. But thank you very much